Hello students, welcome to EPG Pathshala. I am Mohan Chand Joshi, Assistant Professor from Jamia Millia Islamia. Today we are going to discuss about a module, Microscope Based Imaging and Application, Part A, under the paper Genetic Engineering and Recombinant DNA Technology, Part 1. So after completing this module, we expect that students will understand what exactly microscope is, what are the basic structure, principle and the components that are involved in a microscope, what kind of microscopes that are available for them to utilize based upon their applications, and also as we get the details of each microscope in subsequent modules that we are discuss in future slides as well. And in the end, we'll try to summarize about these microscopes. So what is a microscope? Microscope actually is an instrument that allows us to magnify an object. So most of the things which we see through our eyes are in good resolution, in high resolution, but there are several things which we want to enlarge at a time, especially in terms of research. When we're trying to understand how exactly a specific thing is constituted, we require these magnifications, and microscopes gives us that potential, or gives us that tool which allows us to magnify a smaller object into a larger image or larger form. Now, how do we magnify it? So the magnification takes place by using convex lenses. It could be a single convex lens, or it could be in combination of couple of convex lens to amplify or to enlarge a smaller image. Microscopes now utilize light, because if you want to see an object, you cannot see it in the dark. You need to have an object which is well illuminated. Now, illumination of a sample can be done either using natural light, which is available throughout, which is available outside or provided by sun, or it could be visible light, or could be a UV light, depending upon the application which we are interested in, and this will be discussed in future module as well. And there are range, as I mentioned earlier also, depending upon our applications, we can have range of application microscopes that are available to us, depending upon the sample constituent, the constitution, as well as the way it is labeled, as well as the complexity that is involved in a sample. So the microscope can be differ depending upon the complexity or the structure of a sample. Okay, now as I mentioned about the magnification. So here is a table which gives us a perspective how magnification is important in terms of visualization. For example, a naked eye can see a hair. We can see the outline of the hair. We can see a hair which is almost 0.1 millimeter, and that in 1x resolution is enough to understand what exactly hair looks like. But we require a magnifying glasses when you are looking at a plant cells, or when we are looking at an insect, because they are very small and we require at least a magnification of 2 to 5x. There comes the stereo microscope, when we are trying to understand the microorganism, and we require a resolution up to we require a magnification up to 10 to 20x. Then thereafter there are high-end microscopy that comes in, and we are looking at magnification up to 50x, 100x and 200x. And at 50x we are looking at the compound eye of a insect. You know, the insect eye is a combination of multiple lenses, and that lenses we can easily visualize using a 50x magnification. Upright microscope and 100x magnification, we are looking at either bacteria or the paramecium. At 200x we are actually trying to understand how pollens looks like in terms of its structure and its outer outline as well. And then we can go up to 400 and 800 and more x magnification. We can achieve up to 400x magnification and this 1000x magnification using again upright or inverted microscope, and where we are looking at the structure and composition of euglena, or maybe cell or chromosome, and chromosome is almost like 0.2 micron in length, so in width. So by using that higher magnification, we can visualize object of width of 0.2 micron. But anything beyond that we are going to get is via electron microscope, and this technique we will elaborate in next module. But here we can get a resolution up to 0.1 nanometer, and this is very powerful tool in terms of getting to see a very small object in very high magnification. So one of the major factor that is involved in microscopy based imaging is magnification. As I mentioned in previous slide, magnification is the core of microscope based imaging, that we need to get a better magnification. So how does a small object is been magnified? Here is the schematic as well as the details of a magnification process. In a simple microscope is achieved by a convex lens, and convex lens is thicker at the center compared to the periphery, and it is in a simple microscope a single lens is used. So as you can see in extreme right as a depiction of one of the simplest microscope that is available, and magnification via these simple microscopes is almost 300x. That means you can resolve a 1 micrometer small object. This magnification can be improved by when we use a combination of it. So how magnification actually work? So in this cartoon which you see, we have when we are visualizing something from a retina and the sample is on the stage, is kept at the object. So the magnifying lens, what it happens, it amplifies the image or magnifies the image and creates a virtual image which is much bigger than the sampler object. So magnification actually makes a virtual image which is magnified version of a simple object, simple specimen. So because the simple microscope has only one level of magnification, if we have a combination of convex lenses, which is the basic principle of a compound microscope, we can achieve a higher magnification. So the earlier microscope suffered from both chromatic as well as spherical aberrations. We'll discuss about these aberrations in a minute, but the compound microscope, when we start using the combination of lenses, what happens, we can adjust the magnification, and as you can see in this picture, we have the specimen kept at the very bottom, and the angle through which we are looking at the object, instead of making it look like a small object as shown in a picture B, by putting up the convex lens in between, we can amplify it and make it bigger in size. There is a tube lens, there is a tube where the lenses are kept, and lens distance can be manipulated, thereby we can manipulate the range of magnification also. So we can achieve up to 2000x magnification and higher, but this magnification can be lower than 2000x, and because the lenses are kept in tube, we can manipulate that. Now coming back to the uh, aberrations, these are the two major aberrations that are experienced during the compound microscope, because white light consists of different wavelengths, and as the white light passes through the lens, because lens refractive index varies from one edge to another edge, the way it reflects or refracts the single wavelength from a white light varies. Thereby, instead of concentrating all the wavelengths into a single focal plane, it ends up having concentrate different wavelengths at different focal plane, thereby creating a blurry image. Similarly, spherical aberration is another aberration in which we see that light is not concentrated on a single point. In fact, it is on a single focal plane, it's on a different focal plane, thereby creating a blurred image. So this is one of the major challenges that compound microscope earlier faced. Here in detail we are explaining what is spherical aberration is. So in this spherical aberration, because the size and the thickness of the lens as well as the refractive index of the lens differs from center to the edge, what happens, the white light which is coming in is not getting concentrated at a single focal plane, thereby creating an imperfect image or a blurred image. And these aberrations can be removed if we use convex and concave lens in a single in a combination, or we use a spherical lens or a plantic lens that can minimize this one. So essentially what they do, they manipulate the refractive index, thereby bringing in the rays coming from the edge or from the center and merge them into the single focal plane, creating a sharp image. Similarly, the chromatic aberration, as I mentioned earlier also, is because white light is consist of different wavelength, and these lights, due to the different the way the refractive index of the cell of the lens itself is different, thereby what happens that all the wavelengths are not concentrated on the same focal plane, they are at different focal plane, thereby creating a fringe of colors. As you might have seen in pictures where we see the distortion of color at the edge of the camera or at the edge of an image. These are because of the chromatic aberrations, as you can see in this image also, where we have the doublet which are formed, in which the two lenses are fused so that all the wavelengths are concentrated on a single plane. Resolution. Now this term is very is coupled with microscope, and this term defines the capability of a microscope. A microscope is as good as its resolution, and what actually defines as a resolution is the capability of your microscope to resolve two molecules. If two molecules can be resolved, then the minimum distance that is between those two objects is defined as a resolution, and this is defined by an equation where d minimum, the distance between two molecules, is equal to lambda, the wavelength of the light through which that particular thing is illuminated, versus divided by 2Na. Na is a numerical aperture of any objective lens, and because light is a wave, so the diffraction limit is around 200 nanometer for the wave, for the light. That means we can easily distinguish using the very high end microscopy object which are 200 nanometer apart, and it's shown in this graph here also. So if you are visualizing an object, two dots point which are 200 nanometer apart, and if you're looking at the signal distribution of these two, if you try to plot them, you can see both the peaks can be separated out, as you can see in this. But if they are not been resolved, so the graph will be, or the intensity graph will be either a ridge at the peak or completely a single peak, depending upon how far they are. So the resolution limit of a microscope is defined where two objects can be separate out in a two different or two individual peaks, shown in the very first graph. As I mentioned earlier, that resolution of a microscope depends upon the numerical aperture of the objective, which plays a very important role in determining whether what is the minimum distance that can be resolved. As you remember the equation, d minimum is lambda that is wavelength divided by 2Na. So the higher the numerical aperture, the lower the distance between the distance that can be resolved. So the numerical aperture is basically represented as in this cartoon itself, is represents the amount of the light that goes back to the axis, goes back to the camera or the retina. And numerical aperture can be defined as Na is equal to sine alpha, and alpha is one half of the objective angular aperture. Alpha is the angle which represents the one half of the objective angular aperture, and n represents the refractive index. So as you can see, Na in this case is defined if you have a microscopic slide which is kept here, then you have your sample kept shown in red dot here, is covered with a cover slip. Now when the light is emitted from the sample, there is specific light which do not enter into the objective lens, but there are the one which goes back to the lens will also depends upon the immersion oil or the refractive index of the immersion oil if you have. That's why in higher objectives we prefer to use the immersion oil instead of using air, which has a very poor refractive index. Thereby a lot of diffraction happens, and instead of concentrating the signal back to the objective lens, if the signal tend to spread out, and then the angle through which this light can be entered into the optical axis. So here is example of two objective lens with different refractive index, and how this defines the way the signal is observed. As you can see in the lower aperture, lower Na, though you get a sharper image, but the angle is very poor, so the information that is captured from a signal is very poor. At the NA 0.75 you can see a lot of signal is going back through the objective lens. That means that much information is being gathered. So the higher numerical aperture means more information that is getting into the objective lens. Another important factor which is also coupled with the resolution is the point spread function of a objective lens. So the point spread function, as it says, point spread function itself means that in 3 dimension the diffraction pattern of a light emitted by a single specimen. So if you have a specimen and it is emitting light, so how much diffraction is happening in 3 dimension here? As you can see in this graph, it is shown in x, y and z. The yellow line between represents the airy disk, which means there's the point of origin where the maximum signal is there, and then there is a Rayleigh limit. That means from the midpoint of the airy disk to the outer ring through which the signal is still emanating. So in nutshell, or in a simpler language, PSF represents the diffraction that is observed by a single object when you have a light is emitted by a single dot. This is also depends upon the numerical aperture. If you have a good numerical aperture, then you can concentrate back in your signal intensity to the airy disk and get a better resolution. In explaining the PSF in an image, this is an image which is taken from a research article from 2010. As you can see here, that a single object here represented in this heat map, the very brightest red spot in the middle represents the actual signal that is emanating from the specimen. And as you can see, this is in x and z direction. We can see the lateral x and y resolution is 220 nanometer, while in vertical resolution is 520. That means the diffraction pattern for a single point viewed under a microscope will represent a big dot in of 220 nanometer in x and y and a 520 nanometer in a x and z. That is the vertical resolution. This depends a lot upon the wavelength that is being used and what light we are using to or what light is emitted by the samples, also depending upon the refractive index. This particular data in this picture is showing at wavelength of 550 nanometer with the high refractive index objective lens of 1.55. Here in this full width half maximum, that is the Rayleigh limit that we were discussing about, is FWHM represents the difference between two extreme values of independent variables. There are two other important components of a microscope. One is condenser and another is objective lens. So condenser is the one which is used to illuminate the specimen, and this is the one which brings the light from the light source and makes a cone to concentrate on a specimen. And the size of the cone can be varied depending on the condenser we are using. It is very important that we have a proper illumination so that we maintain a cone shape structure through which a cone shape configuration in which the light is coming and hitting the sample. If it is not maintained, the sample would not be uniformly illuminated, resulting in a poor contrast. Similarly, objective lens is the one which can be a combination of various lenses. As I mentioned in my very first slide, that compound microscope they use combination of two or more lenses, and here is an example of it, that nowadays objective lenses are in combination of up to 15 lenses which are arranged in different ways so that there is minimal chromatic aberration, as well as the maximum light is being taken from the sample and used for image projection. These objective lenses are nowadays well known are achromats, fluorides, apochromats or plain apochromats. These are the terms which are used, and based upon the amount of correction each of these objective lens are. They are named very famous and very high-end objective lens are mostly plain apacome matte. And in this figure here, we can see the condenser is at the bottom, the specimen is kept at the stage, and as you can see from the source of illumination, the light source, condenser's job is to create a cone and hit it to the sample, and that signal is captured by the objective lens. Illumination. So condenser takes the light from the illuminator or the light source and projects it to the for our sample. But the degree of illumination is very critical, is because a uniform illumination is very crucial in getting a high contrast image. And the illumination intensity or illumination depends upon following factors. What is the depending upon the following factor? One of them is the intrinsic brightness of the light source, the focal length, the distance that we have between collector lens and the source, condenser numerical aperture, condenser aperture diaphragm, and overall transmission of illumination system. So these are the things in combination which are very important for illumination. What are the sources through which the illumination can be done? They are tungsten lamp, then you have halogen lamps, mercury lamps, xenon lamp. Nowadays metal halides and the LED are the two most famous ones that are used for the fluorescent based imaging approaches. So in all of these cases we generate a white light, and that white light or light is generated when we have a wire. Especially in case of the lamp that we are using, we have a wire which is surrounded by inert gas, and there is an arc which is produced which results in a white light, and that white light can be directed toward the condenser. Similarly, halogen lamp, mercury lamp, xenon lamp are using a different combination of inert light and the arc to generate the light. So detail about one of the illumination source which is commonly used nowadays is metal halide, and metal halide is actually a discharge lamp that is located inside an elliptical reflector which focuses the output light through a liquid light guide before it get transmitted to the microscope. And as you can see in this picture, the lamp base, and you have the arc which generates the light, and then you have the elliptical reflector which concentrating the light toward the thermal filter, and now through a liquid light guide that light is eventually been coming out to the going to the scope. So the intensity is maintained or can be modulated by the focal length through which the distance between the reflection and the arc. The latest one, the recent light source are the LED one, which are similar to the conventional illuminator because they produce a range of produce a broad range of white light. But the advantage with these ones that we can provide filters which can remove a specific wavelength of our interest, whichever we want, whichever we don't want. We can put a filter in front and then make sure that to our system only specific wavelength is being discharged or given. This is very important in terms of our imaging, in fluorescence imaging in particular, where we don't have to worry about getting an excitation filter before the sample is getting hit by a light. And in this graph we can see the wavelength that are available. So the red represents the tungsten light, and with the relative energy we see the tungsten lamp produces energy, produces all the wavelength but has the higher energy at the higher wavelength. Mercury vapor lamp also have a range of white light, but the intensity or the relative energy peaks at specific wavelength. White LED light on the other hand will have more or less relatively same energy except higher at the 488 region, that is the green region. Apart from that, it has a very similar profile in terms of energy. And the noon light, the sunlight, will have a uniform distribution of all these wavelengths with relatively equal similar energy. Therefore LED provides a very good highly reproducible light source. Here is the brief description of different types of illumination. What are the intensity that we are getting, what are the luminous flux that we are getting from the sample, and what are the spectral range that we are getting. Again, we see that LED has a better value throughout, and halogen or the tungsten light, the very first that one used, at a very high radiance flux as well as the luminous flux, and provided the very wide range of wavelength. Similarly, mercury, xenon, and metal halide as well. So here is the overview of, or a brief outline or the schematic of a fluorescence or microscope. And in this, what you see, you have a stable frame, which is the reverse L-shaped frame. Then you have the light source plugged in on the one end. It could be tungsten, halogen, LED, anything. So which throws the light, and light passes through condenser and then hits the dichroic mirror, through which it has gone to the objective lens, illuminated our sample, and when the sample is illuminated, the light is transmitted back, dichroic mirror as well as the filter, emission filter, and then can be seen through the eyepiece. Also, you will see there is stage, mechanical stage, which can be used, which can be rotated and can be moved in X and Y direction so that the sample can be placed. And there are various other features which are shown in this picture as well. So there are types of microscopes that are available depending upon the application. For example, binocular stereo microscope, which has a lower magnification, but what it allows you to observe objects in 3 dimensions. Then we have a bright field microscope, which is the standard microscope, which is standard form of light microscopy used to visualize objects. So you can use to visualize live cells in it. Then we have polarizing microscope, phase contrast microscope, DIC, and fluorescent microscope. These will be discussed in details in next module. Also, we have the high-end application of these microscopy, where we have the total internal reflection microscope, very important and very crucial to for the imaging of membrane. Then we have confocal microscope, we have 2-photon microscopy as well as multi-photon excitation microscope, and we have SIM, structural illumination based microscopy, to get a higher resolution and the more information about the sample composition. Okay, so the type of microscope, as we mentioned earlier, there are 2 different kind of microscope here: electron microscope and a scanning probe microscope. These 2 microscopes are very different than what we mentioned, than are the one from that we discussed earlier, and will be discussed in detail in a separate next module as well. So electron microscope basically utilizes electronic beam as a source of illumination compared to a light source or a UV light, and they have 2 variants, either transmission electron microscope or a scanning electron microscope. What happens in both the cases, the electron beam which is generated by electronic gun or electron gun is accelerated by a via anode, and by using the electromagnetic lenses, they are used to target it on a sample. Now sample will either illuminate, will transmit the light, and that signal will be used to generate an image, or it could scatter depending upon the way it interacts with the surface and the complexity of the sample. So those secondary electrons which are, or the scattered electron which are resulting due to the surface, are being captured and used to generate the image. The difference between these 2 techniques, TEM and SEM, is the way they generate 2-dimensional data or the 3-dimensional data, and this will be discussed in future slides and future modules. The second is scanning probe microscope, where instead of using either electronic beam or the lights to illuminate the sample, in this the sample is probed physically. For example, in atomic force microscope, you have a cantilever where you have where you are actually tapping it on the sample, and whenever there is a contact between the probe and the sample, this information is captured by the computer, and then that information is used to generate a three-dimensional map, because it continues to tap and also look at the surface at the sample, and whenever there is a contact, that information is registered. And on the other end, the SNOME is also based upon the interaction of the evanescence wave that are generated once the specific wavelength light is passed through a aperture smaller than the wavelength. It generates a evanescence wave which resonates in that area and also gives information about the sample it is interacting, and that information is used to generate a high resolution imagery. So these two techniques are very different from the light and the fluorescence based imaging microscopy which we discussed earlier, but gives another dimension to the application that are available with the range of the kind of microscopy we have available. So I hope the students have understand by the end of this module that microscope is become a very integral part of researchers in biomedical or the basic researchers, because of the range of application it has. It has given researcher the potential to expand and to understand the biological complexity by using these techniques, by using this microscope tool. Microscope, as you can see, has also evolved over the years. To begin with, where the light source were just the natural light or the mercury lamp, from there it has evolved up to two photon system, where we are just using very small amount of light to illuminate our sample to get a better resolution. The basic component of a microscope, however, remains the same. It requires that you have a very high quality objective lens, so that we have minimal aberration, whether it's chromatic or the spherical aberration. Also, the source of illumination has also changed, whether once we had the natural light to the arc lamp, now we have high quality LED light source that are used to illuminate our fluorescent tag. The magnification and the resolution of a microscope remains the hallmark of microscopy imaging, because these two things has to be high, should be good in terms of addressing any questions, and they both depend upon the numerical objective of the objective lens. So that is very important for microscopy. And then, as I mentioned, that as the technology is advancing over the period of time, the new modifications has been adapted by the microscopy to give you better result, better reproducibility, and better application in the biological research. Thank you.